And now I want to introduce our very distinguished Dr. Sue Griffin that you've seen kind of under and around me up here. We're getting ready. And Dr. Griffin is a professor and vice chairman for research at the Donald W. Reynolds Department of Geriatrics here at UAMS. Her three major interests include molecular biology of Alzheimer's disease, and her research interests focus on mechanisms in the progression of Alzheimer's disease. And she recently discovered a link between the presence of a certain immunoresponse-generated cytokine, interleukin-1, and the increased probability of onset of Alzheimer's disease. This is seen as a significant breakthrough in the early detection and treatment of Alzheimer's and has drawn national acclaim for Dr. Griffin and the Reynolds Institute on Aging and Department of Geriatrics. Today she will be speaking on the future direction and research update on Alzheimer's disease. So my longtime friend and colleague, Dr. Griffin, welcome.
Now, this is a list. You got it, I think, in a handout. And I just want to tell you that, of course, this is science talk. This is scientists talking to each other and, you know, doing it by these bullet points. But today, of course, we're home. And so I'm just going to tell you that when I get through with my talk, then you'll know all this so you can, you know, tell others. Okay, let's talk about when you're old but okay. And old but okay means that you feel good. And you know, people that work in aging don't really prefer that you call it successful aging. But if you can live to be old and you are not demented, that's success. And you know, you may have a few other creaking parts and you may be having some other problems, but as long as you're not demented, it's a success. And one of the things that I think people, when they are 40 and you're talking to them up on the floor to tell them what our interests are and what we're working on, you know, heart disease, that's Dr. Way's big program, and falls and frailty, that's Dr. Sullivan's big program. And these things are major concerns of people when they're thinking about getting older. But the top thing is I don't want to be demented. I want my body to go at least at the same time my mind's going, if not before. So these kinds of issues then, we have to think about them not only as we get older, but as we see our children get older. And because we want to stay healthy enough so that we can make our own decisions about things that we can do with our brain. So here's this old guy, and you can see he looks like he's bored stiff. And you can imagine him in his Barcalounger, and he's watching TV. And when he's watching, he sees on the screen this person that reminds him of one of his old girlfriends. And suddenly he remembers everything. You know, he saw this with his eye, and it went into the brain, into the area of the brain that you have a picture, you know, a snapshot is made, so to speak. And that visual image then is carried by neurons, and I'll show you how this happens in a minute, neurons that then connect to other areas of the brain. And those other areas of the brain are ones that make associations. And all of this is happening at breakneck speed. You know, when the person behind you at the stoplight and you don't jerk the car forward and they beep, you think of that as a millisecond. That's not a millisecond. A millisecond is 1,000th of a second. And so I just want to tell you that that is the way the communication goes on in your brain from a neuron to the next neuron. And so this information that he's seeing with his eye, at the same time he keeps watching the film, that information is carried to association areas of the cortex and also down to this area that's so important in learning and memory. That's the temporal lobe. And that area is learning and memory and old memories. We don't know exactly where the old memories are, but this is the conjuring in your mind, and you've done this, I know, yourselves, where you actually, you get the perfect image of the person that you're reminded of, and you get everything else with them. You get the way they smelled, you get the way they felt, you get the way you felt when you had your hands on them. You feel everything. You see everything. You imagine where you were the last time or the first time. And so this kind of memory is the kind that we can snap back anytime we want. And one of the luxuries, even if you can't remember whether you had green beans for supper last night, you still have these old memories, which are a comfort. Now, this is what the brain looks like even in an old person that's cognitively intact. And you can see these areas. The yellow ones are the most active. And that little thin layer where you see the yellow, that's the area of the brain that we think with, that we have every imagination we have every motion that we want to make, and it says, okay, do that thing. All of that is contained in a little tiny sliver that's maybe 1/4 of an inch max. That's where the nerve cells, or the neurons, are. And all of the rest of this is big pathways that go from one place to another, that carry the information from one neuron to another neuron. Okay, so I just want to tell you that this is just a small slip of cells. It's like this slip that you put on a pillowcase, on a pillow. And you know, that little bit is what's running the show as far as we're concerned. You can live, but you won't be thinking, and you won't be having ideas. Okay, so now that has introduced you to where those nerve cells are. And that also should introduce you to how fragile that we can do everything that we do. And so we really need to take care of it. And at the end, this is not very many slides, so I wanted to leave a lot of room for us to just chat about questions that you all have and the questions that you all have at the sites. And here's the left brain. This is my favorite picture. You can get all kinds of pictures off when you go on Google and look at images. And this one I love. I'm the left brain. And I have one son that is left-brained. He's a mathematician, a theoretical mathematician. But he also, thankfully, you don't just have to have one side. You have two brains. And these two brains then are working together. So the left brain for your logical thoughts, like, you know, got to get my tax stuff ready for the CPA this next week, for sure. Should have done it last week. We'll do it next. Okay. And then the right brain, that's where you have love and passion and freedom and the things that you really want as you get older to really develop. I mean, we should be able to enjoy old age, especially if we're successful. That is, we don't have problems with our ability to think, do, and make our own decisions. Okay, this is how it works. This is a nerve cell, a neuron, as I want to call it, and it is speaking via this axon, and that axon is carrying information. It's carried electrically, so it's like we have a plug-in in the brain. And so this is the wire that the electrical signal is coming along, and at the plug-in, this is called a synapse on cell number 2. Okay, this synapse, the electrical signal sends the impetus for the release of these little red neurotransmitters. So it's transmitting the news that neuron 1 got to neuron 2. And that little cleft is so important. And it's not the only thing that's there, and I'll show you that in a sec here. Okay, there are also these cells that act as helpers, like nurses, and like people who are standing there helping you out if you get in trouble, or even if you don't get in trouble, just to make you more effective. And so here, and they're called glial cells. Here, this glial cell is wrapping 2 of its processes, and there are thousands of these happening, but this is just to make it simple. Okay, so these glial cells, then they are releasing these small factors, or cytokines, into the cleft where we saw the synapse, releasing it right into here. And do you know what it does? It's the most amazing thing, and we didn't know about this 10 years ago. So when they release their transmitter into that cleft, it modulates what neuron 1 can say to neuron 2. And here it's just showing that the thing it does here is to make more of the receptors for the neurotransmitter. So you get more information passing between neuron 1 and 2. And there are other ones that dampen it. But it's modulating what it says. Now here's an experiment, and I want to introduce you to this because I think people need to know about science. And I don't know how many of you have children or grandchildren, but this is the thing that you can give them. You can tell them that, look, if you can lie, I guess. It was so interesting today. We saw data from real experiments. And in that way, maybe let them understand that you appreciate so much what they try to do to have more power in their life, more math, physics, science in their lives, because that's what helps us in our daily life to decide, does that sound true? Or does that sound wrong? Does that add up? Or does it not add up? And those are the kinds of abilities that you want, I certainly do. I want my children and my grandchildren to have that ability to make decisions. And that's what it's all about. So this is a way of making a decision. Because one of the things we wondered is whether or not if you treat a neuron, and this is done, of course, in culture, and that means it's in a glass, old times, plastic now. So you put the neurons in the dish, and you then treat them with something that'll give them a little bit of stress, but it's the same stuff that excites neuron 1 to say something to neuron 2. And that's glutamate, this stuff called glutamate. And you see that sometimes in the 2 or 3 inch ingredient list on foods. But anyway, here's glutamate, and it's exciting, stressing a little bit, these neurons. And the first thing that happens is there's an increase, just right increase, in a protein that's necessary for maintaining the neuron, maintaining the health of it, maintaining its membrane, so its membrane doesn't break open and then the cell dies. So it's necessary for that that process to occur. So here we see these are the cells that didn't have glutamate on them, and you can see the difference between the amount of this BAPP, the beta amyloid precursor protein, which is the precursor for the amyloid plaques in the brain. And here you can see at the same time, outside the neuron, it releases this secreted form. So the beta amyloid precursor protein that's put in the membrane, some of it sticks outside the cell, and it gets cleaved off, and it's this, SAPP. And so what that means then is the increase in the amount of APP that was made because the neuron was stressed a little bit or caused to fire and send the message down. And at the same time, there's the release of this SAPP. This was a discovery by Steve Barger, and it was so important because it was the first time we knew how a neuron that's being stressed speaks to its glial cell that's trying to help. And this is it. This is what happens. So when you have this SAPP, you put these two cultures together, the neurons on the top and the microglia, one of the glial types, in a dish on the bottom, and you put a membrane between that only small stuff, tiny actually stuff, like this SAPP, can go across the membrane to act on the microglia. And this is the situation where it was glutamate treated. So you can see the increase compared to the untreated. You know, this is the treated and the untreated. And just to make sure, we put on the glutamate at the same time we put on an antibody that would attract the SAPP so it could not act. And here you see it's just like the untreated. And what was amazing about this is the little cytokine that Robin was talking about it when she was introducing me. This is the cytokine. It's called interleukin-1, and we just abbreviate it as IL-1. And there's two forms of it. One's an alpha and the other's a beta. You're going to be scientists by the time this hour is over. And not only is it more made in the cell, more of it's released. And it does so much stuff. It causes an enzyme to be made that breaks down the neurotransmitter that your brain is trying to make for acetylcholine, which is so important for learning and memory. And so it causes it to be broken down if it's in excess. But if it's just right and your brain is okay, then you have the amount of acetylcholine that you need. But when you fire the neuron, some excess goes outside. You want to get rid of that because you don't want the neuron to keep firing, right? Okay, now that you're a scientist, we can go on. I'll show you what this looks like in a cell that's stressed. And this is a human cell, and this is from a person with epilepsy. And epilepsy is so much like our experiment in the dish because it's glutamate that's overexpressed here and that's causing the firing of neurons in epilepsy. So in epilepsy, you have a focus of nerve cells or neurons that are firing. And because they are firing, they're releasing their neurotransmitter. And that neurotransmitter is speaking to other neurons all over the brain in a person who has epilepsy that causes them to do the whole epileptic event of, fall down, etc. And here, what happens, this red is colored. Oh, this is so amazing. 20, 30 years ago, you couldn't do this experiment. You couldn't look at the brain like this. And what this is saying is this neuron is stressed, which means, because now you're a scientist, it means it's making more APP, because it's under stress, and releasing more secreted, or SAPP. And that secreted SAPP is activating and causing the increase in expression of its glial cells, and these kind of glial cells just smack right onto the neuron. You can see clearly in an electron microscope, but not in a microscope like this. So you can see they're just slapped up against each other, it seems. And the SAPP has been released. The microglia, this is a microglia, is activated, and it's making a ton of IL-1. And the reason I can say it's a ton, in quotes, is because the microglia is so big and has so much, here shown in yellow, and it's in the intensity of it gets to be brown, so much of it is released. Now this causes this neuron to have some things that are better off, because in these kinds of situations, not only is the big neuron where you see all the red, but all of the processes are affected from this neuron. So that means at the synapse, you need to make more of the proteins that are necessary for repair of the synapse, that little cleft, and the neurotransmitters. Every one of those things is regulated by IL-1. So when IL-1 goes up in the microglia and it's released, then in this neuron, those proteins are, the neuron is responding by increasing the production of those proteins that are necessary for the synapse to be repaired. And you know, these stresses are very hard on neurons, and if they're intense enough, the neuron dies. So help with the repair of that synapse, help with repair of every aspect of the changes that occur because you have stressed this neuron. Now I want to turn to something that's a little bit more of a good news thing. That was good news because the neuron will not repair itself without the cytokines. I'm sorry, you're going to be scientists when you get out. But I want to show you what it looks like in a person who lived his whole life and didn't have dementia and didn't have any of the changes that you can see in a microscope that are like Alzheimer's or Parkinson's or any other kind of disease that neuropathologists recognize. Okay, here's a person that is 16 years old, and these two proteins that we're looking here are necessary for the proper function of the neuron. So this one, this is IL-1. You can see that it's kept very low as long as nothing is happening. And then when you get to be about 60, it goes up a little bit. Nothing like Alzheimer's, but a little bit. And that's so that the stress, I don't know what all they are, but we know a lot of them, that is put on a neuron just by getting old. And so this is a picture of that. Here, this was about 60, you have this increase. And here at 59, you can see an increase of these two proteins. The APP, just like we almost knew before we looked that it was going to be elevated, because that's the stress response, right? And this, dynactin P50, is very important because for the APP and these other proteins to do their work, they have to travel inside the cell. So they have to be transported. And this thing right here, dynactin, is the transport vehicle. It's the car or the bus. And you can see that as this APP goes up, then the transport vehicles also are increased. And everything is fine. This person died without neurological disease. And we do all of our work in humans, of course, on autopsy material. And here you can see the difference in that in Alzheimer's disease. And that's what we are studying right now, is a real exploration of why does this happen. Here, instead of being in the neurons and transporting these proteins around where they need to be to do their job, in Alzheimer's disease, they get on the bus, but then they're stuck. And these proteins that are necessary for repair of the membranes, repair of whatever is happening in the cell that's untoward, and they all, instead of being able to travel around and do their job, they are stuck. So the vehicle has them on the bus, and they're stuck. The vehicle's stuck, and they are stuck. And the stuff that should get rid of them is stuck in there too. And that's why you see these neurons that are full of these proteins that need to be gotten rid of, they're used up, or they're misfolded, something's wrong with them, and they need to be got rid of. And whereas this person and the 6 other ones we did had a picture like this in successful aging, but in Alzheimer's disease, they have a picture like this. So here, they're traveling around the cell, able to, and do well. And here, all of it gets stuck together. It gets stuck in the cell body, in the neuron cell body, and also in the processes that go from neuron 1 to neuron 2 and neuron billion. So that's the trouble and the difference. But that is so clear, and we feel that since the difference is so clear, we will be able to tease apart these changes that are happening, changes in the vehicle, changes in the proteins that are supposed to get rid of them because they're spent. You know, they've already been used. They're used, so we don't want them anymore. We may want to just totally get rid of them, or we may want to recycle them and make new proteins with the amino acids. Okay, now here is what that protein I was telling you about that's necessary to get rid of them in the neuron. Well, here it is. So you can see clearly that it is stuck as well as those other proteins. This is Alzheimer's disease. This is Parkinson's disease. Same thing. In Parkinson's disease, the real problem molecule that's made in excess is this little protein called alpha-synuclein, and it's necessary for maintenance of the synapse. There are many of these kinds of proteins. I'm just showing you this as an illustration. And this p-tau is necessary for the transport, but it's overexpressed, and when it's overexpressed, it gets stuck as we show here. It gets stuck with the APP, with the bus. It gets stuck with anybody that wants to take a ride on the bus that can misfold or aggregate. And so here, I'm showing you the examples of that. Parkin is the little molecule that's trying to help them get over to be recycled or got rid of. And here in the normal, you can see there's a very nice distribution of all of these proteins that are reminiscent of what we see here, a very nice distribution within the neuron. Everything is fine. And here you can see where they are, these things, the bus and the proteins, the alpha-synuclein and the phosphorylated tau, they are all stuck together with this protein that labeled them so they could be got rid of. And so one of the things we are now beginning to study is what is wrong with, of course, the process they haven't even got over, but there are entities inside cells that are used to recycle or get rid of unwanted proteins. Now here, this is a study, and this is a little bit of a jump, but I want to tell you this because it's aimed directly at this molecule, this cytokine, that is inducing or causing to be expressed in excess these proteins. So this little interleukin-1, which is the interleukin or the cytokine that's inducing that, both these things, p-tau, alpha-synuclein, and even Parkin. So the cell is trying to do everything it can to help itself, but it's just not working. But this is in a mouse study from Dr. Craig Cole. So this was a study done by him in mice, and these are mouse models of Alzheimer's disease. So the way they model it is they get plaques. They get these aggregations of A beta. They also get, if they're the right kind of model, they also can get aggregations of the p-tau. And we have another one that's for alpha-synuclein. And we'll be using all of those in our studies. So in the transgenics, he compared the two transgenics. This one did not have ibuprofen treatment. And you can see here in the controls, and then when you look at the transgenic that was a true transgenic. I'm sorry, this is mislabeled. This should say wild type. He calls it transgenic minus. It's not a transgenic. It's a sister or a brother of a transgenic. And this one is the transgenic. So in the transgenic, he has a lot of expression of IL-1. But when he treats that transgenic with ibuprofen, so they're given ibuprofen in their food, and you can see that the amount of IL-1 that's made is dramatically decreased. And that means that the overproduction of the APP, the overproduction of the p-tau, and the overproduction of the alpha-synuclein, all of those things are going to be controlled. And the way he looked at these animals was to show that they had fewer of those plaques that contain the A-beta. And that's a hallmark of Alzheimer's disease. So just with feeding them ibuprofen. And now I want to show you an epidemiology study. And this came from the VA database. The VA database is like a fountain of information. And that information is so virtually perfect. All databases have problems, or have, differences and problems. But in this database, the way you get the database is you record what you do with every patient. So if a patient comes in and they say, my knee is still killing me, my back is still whatever, then at the VA, because it's socialized medicine, so to speak, at the VA, then of course they start with ibuprofen, which is really good, or naproxen, or something cheap, aspirin. And so these people were treated for whatever their aches and pains are. And one of my nieces has a sort of benign brain tumor, and so she has headaches a lot. And of course she's offered every drug to help her headaches. Ibuprofen happens to be the best one for her. So, people respond differently to whatever kind of medicine there is used with them. But it's always recorded in these vets that come. And that's put into the VA database. Not only that, the person has to go downstairs in the VA. You can go look yourself. They go downstairs in the VA and pick it up. They pick up ibuprofen from, or whatever they're taking, from the pharmacy. That's recorded in the database. And they have to come back to get another one. And that's recorded. So it's an excellent database. Now what they did here is they took 50,000 people who had died with Alzheimer's and 200,000, well, I shouldn't say they died, they had Alzheimer's disease, because this is a database of living people. And so they had 50,000 had AD and 200,000 died without AD. And so what the study was showing is, what's your chance of being in the AD group versus being in the not AD group? Well, this is your chance. So if it were just equal, could be one, could be the other, which is where you start anytime, that's 1. And then if your odds start going down, that means you're less likely. Every downward movement, you're less likely to be in the Alzheimer's group and more likely to be in the non-Alzheimer's. So, what do we see here? This is up to 1 year, up 1 year to 2, 2 to 3, 3 to 4, 4 to 5, and 5 years after 5 years. And people who have these kinds of pains, like my niece, they are chronic users. They're not stopping. And if she were getting it from the VA, she'd be going over there and getting it instead of going to Walgreens. So in the group, so it's 250,000, If you'd been taking ibuprofen, it would lower your chances of being in the Alzheimer's group to almost 50%. And the other drug was naproxen. And this group also very good. I mean, 25%. If we could do this, 10% fewer chances of being in the Alzheimer's group, it'd be just fantastic. I mean, people would say we don't have Alzheimer's anymore. It's so dramatic, and people, because we're human, we think of one case than this other case, and we think of our grandma and somebody else's grandma, and you know, we don't think about it in the large realm. But in our country, taking care of Alzheimer's people and Parkinson's and other people who have kinds of diseases that can lead to dementia, that's costing us the most money, period. We are spending a tremendous amount of money on taking care of these old people. You know, everybody wants them to have care, and that's wonderful, and I'm glad we have still the resources to do it. But I'm telling you that if we could lower the risk of it by any one of these things, at any point, it's worth doing. So now I want to ask you this question. Why on earth are we not having something that tests this out in a group of patients where we know what they've been doing since they came home from a war? So that's one of, and I made this talk to, and that's why I had the first slide there. I made this talk to a group of scientists at the VA. And so now we're trying to put together a study that will allow us to test this out in a population that we can really control. And now there has been a study done looking at these, whether or not this could be done in a setting. Now they haven't used ibuprofen in the setting, but they did use some drugs, and one of them was celecoxib. They had stopped the trial because celecoxib started making problems in the patients. And that was something that all of us could read about in the newspaper when the celecoxib was, you could still get it, but it just was not going to be done in that study because people were being affected. So, you know, this is not attractive to companies to do, and I understand that completely. I really love business, and any business that I get to do is so minor, farm business, but I think it's very important. Everybody wants to make money, and that's very good, but this is a public health concern that's concerning every one of us that may or may not be making any money to speak of. And so I'm trying to get a study like this going in the VA with VA researchers who have access to patients and have access to this database. So we already have, the news is in, that in people that had been taking it, that their chances of being in the Alzheimer's group is dramatically less. Now what we need to do is see if you could treat people for 4 or 5 years with ibuprofen. You can try all of them you want. At the VA, you have the patients that take X, Y, and Z. And look to see whether or not we could repeat this epidemiology study, even to an extent. And we have people that are already being treated with ibuprofen, so we have a jump on it. They're not dead. They've already been taking it for 5 years, so we could begin to study that information that we have gained on them and then check to see what happens over time. Because the database, you know, if you live to 96, and I've seen the stacks, they are huge with all the records that you get. But if you live to 96 and you're involved with the VA, you're still being in there, put in every time. So we could actually get information that's just as powerful as this with our own patients. and when I say our own, I'm also appointed at the VA. I don't see patients because I'm not a doctor, but I do see the data from them, and I do see the need for doing this kind of study. Now, I want to stop there, and I want us to have time to talk and have any questions that you want to ask, anything you want to talk about, then I'd be so happy. And I'm hoping that the sites are going to be joining in.
I know in the aspirin study recently they said that the enteric-coated aspirin did not work as well as the ones that weren't coated. And so is that true, do you know, on the ibuprofen? And also, I know since it's not been a controlled study for Alzheimer's, do you have an idea of what the doses are? And is there something that protects, a way to take it that protects from the side effects of the GI upsets?
Yeah, you know what? One of the things that, that's a very important question. But I want to say two things about it. One is these people in the VA who were taking ibuprofen, if you went back to the database, you could find out just exactly to a milligram how much they were taking and what kind it was. So it's in the database. And so, but you have to mine it. And now every day, the database is more and more secured. That means secured away from research, which is very unfortunate. But that can change, and it will change. It just has to have someone who says, we have to do this, we have to look at this. And it's so important, because you know what it means? All these guys that are coming home with PTSD, that absolutely bombs the area of the brain, the temporal lobe that I was telling you about. It bombs it. It makes it even look small on when you do scans of the brain. So just imagine all those guys. We heard about one in particular at Fort Hood yesterday, day before. Think about all them coming home with PTSD, and we're not doing anything that could possibly help them, except by accident, ward off, prevent, delay the onset, or whatever you want to call it, of getting Alzheimer's. I mean, so you have to live with PTSD till you're 60, and then you get Alzheimer's? Or you have to live with brain damages that have happened to you by blasts or direct trauma, and then you have to look forward to getting Alzheimer's? No way. I mean, that we can't, that's wrong. And besides that, you know what? We can't, even as rich as we are as a country, we cannot afford it. Because there's thousands and thousands of them. And you know, if you're just a person that didn't ever go to war, your risk is something, whatever it is. It may be zip or 2% or whatever increase, but there's a risk, and a big risk now is CTE, chronic traumatic encephalopathy. It's talked about for football players, and honest and true, it's beginning to be talked about for soldiers, but not much. And one of the bad things that, could I just diverse for just a moment, digress, what they're really finding out with football players is it's not playing the game, although that can be really bad because lots of concussions happen, but it's really the practice, especially in 10-year-olds and 11-year-olds, and these kids that are being put out to play this game where, I'm sorry to report, coaches want them to play like they're in the NFL. You know, they want them to block each other, so they hit each other as much as their little bodies can create an impact. So now they've found out, amazingly enough, that that's it, that it's the practice, and especially in kids. So I don't know what you think, but I know what I think. I thought my kids were so incredible, and they were, that I had to keep hold of them most of the time, and if they got out of my sight, I was shouting for them. So I don't know. Yes?
But
Oh, I'm sorry, I can't quite hear it.
I said, I think you've addressed this a little already, but, okay, what is the research that's been done for chronic stress that causes memory issues in relationship to Alzheimer's? Am I making it clear?
You mean the chronic stress of what? Like in your life? Oh, well, I think there's two things. Some people have disastrous things happen to them, and they respond by coping, and they go on, and you know, so their precious husband dies or their precious wife dies, and they don't give up, and you have plenty of stress, and so then people say, oh yeah, but you know, they're just different. Well, actually, of course they can be different, because you know, we all are genetic products of whatever we got from our mama and daddy, but they can be different, but also they can be helped be different. And that's why it's so important when you have a lot of stress to talk about it. And you know, I don't ever like to, nobody does. Nobody wants to spill the beans to somebody that they think is going to carry it all over the building or all over their community. But you need to find somebody who doesn't talk, and you need to talk with them and let them know what you're doing. And they may be telling you back something that they went through that you didn't even know. Or maybe they'll just be telling you, oh, hang on, come on, don't leave you alone until you come and go with them to whatever. And another thing that's so important, and if you have a lot of stress, stress can be bad, but then I could have another lecture talking about what it does to the hippocampus to be stressed, because you release a lot of cortisol, and you know, you have a lot of stress put on the neurons there. But you know, stress, that's how come we stayed alive. I mean, we wouldn't have got out of caves hunting mammoths if we hadn't been, you know, if stress hadn't been partly a good thing. And so we are made to deal with stress. Now, every person is different or somewhat different, and maybe some people can do it better than other people. But everybody can learn to some extent to cope with it and not let it ruin the rest of your precious life. And you know, sometimes it's hard to see how precious your life is when you've gone through some loss, or your kids are not turning out, or they're coming back home, or whatever is stressing you, or you're having to take care of somebody who's totally demented and doesn't know you unless to shout at you. And so, you know, all of these, oh, I have to mention this. At our center, we have a place where you can come and learn how to cope with it if your problem is caregiving. And that's the program, right down on the floor where you enter, on second floor. Come to the middle elevators, and it's right there on your left, and that's Robin's program. So there are ways and there are places where you can go that will help you if your stress is starting to overwhelm. And anyway, it's just what we ought to do for each other. We ought to be there for each other. And so I'm hoping, and I wish so much that the other sites, if they had a question or anything, would chime in. But I just want to tell you that it's so good to talk with these sorts of, talk about these issues with a group that we can take from, oh.
There you go. I wanted to see you.
Oh, okay. Hello, everyone out in the sites. I hope you're still hanging on. But anyway, yes? I heard some.
My question.
Yes?
Can you hear me?
Yes, fine.
Okay, my question is about the Alzheimer's in Down syndrome. Are there studies being done on that?
Yes, thank you for mentioning that. Yes, that's part of our study. We have a collection of tissue. We have about 56 cases. We have a collection of tissue from people with Downs. And, oh boy, I hope nobody's recording this, but Downs, if you could possibly just take this point of view, it's like a model for, it's like the model, you know, we talked about the transgenic models. Well, this is like a model of Alzheimer's disease. So you see the early events, and that was my original discovery that said this overexpression of interleukin-1, we showed it in Down syndrome. So it must be an early response to neuronal damage and starts a cascade that because it causes more damage, then the glia respond more. So it creates a cycle. So yes, and that is part of our new proposal for our program project. So thank you very much for saying that. Anybody else?
I have one announcement I need to make.
Okay, all right, great. And thank you so much. I want to tell you that my phone, I'll even give you my cell phone. So if you want to call, it's 501-766-6530. And I get calls from far-off places asking me these same kinds of questions. So if you want, you can call me. And if I can, I'm going to answer. And if I can't, then I'll get back later. Thanks.
Thank you, Dr. Griffin. That was wonderful.